Hello everyone, welcome to the new session of Biomolecules exercise. So today we are going to solve the questions from the NCERT, right? From the chapter Biomolecules. Okay, so let us start the session with the first question. The first question comes here. What are macromolecules? So there are two types of biomolecules. The one that is called as macromolecules. Okay, and the another one is the micromolecules. To give examples of the macromolecules also. So let us see the answer. So the macromolecules are the large complex molecules that occur in colloidal state in intercellular fluids. So there are intercellular fluids in between the cells. There are some fluids filled. Okay, so these are some of the macromolecules that are present in the colloidal state. Okay, and that is in the intercellular fluid. Now they are formed by the polymerization of low molecular weight micromolecules. So there are small molecules which are called as micromolecules, and their molecular weight are comparatively less than the macromolecules. So what happened? The micromolecules get polymerized, right? They are under uh, polymerization reaction. So these micromolecules are considered as a monomers, and when monomers combine together in the form of polymerization, they are now called as macromolecules. Okay, so macromolecules are quite larger than the micromolecules, and here the micromolecules are acting as a monomer of the polymer, right? So by the process of polymerization reaction, these micromolecules unite together to form a polymer like structure, right? So polysaccharides, okay, polysaccharides are the polymer, and they are made up of the micromolecules, that is the monomers. Okay, similarly the proteins are made up of amino acids are joined together to form the polypeptides, and different polypeptides unite together to form the proteins. Okay, now similarly the nucleic acid. Okay, nucleic acids are also the examples of the macromolecules. So nucleic acid, if you see, there are the monomer is the nucleotides. Okay, these are the nucleotides, and nucleotides unite together. Here the nucleotides are micromolecules. Okay, now nucleotides unite together and form the polymer of nucleic acid, right? That is the DNA or RNA. So macromolecules are the large complex molecules, where the micromolecules are the simpler form of the biomolecules. Now, the next question is, what is meant by tertiary structure of protein? If you see the structure of protein, then they are of different conformation. There are a primary conformation, there is a secondary conformation, there is a tertiary, and then there is the quaternary structure. So here the question is about what is the mean by the tertiary structure of the protein. So the helical polypeptide chain undergoes coiling and folding to form the complex three-dimensional shape referred to as the tertiary structure of protein. So it means that there is a primary structure. First of all, there must be a primary structure of a protein. So what is the primary structure of protein? First of all, let us understand that. So primary structure of protein is, you can say, the amino acids are working here as a monomer. So monomers, amino acid working as a monomer, they join together and form a polypeptide. Now this polypeptide is showing the primary structure. Okay, now this primary structure that can get folded in of two form, either of the two form, that is the alpha helical structure or the beta pleated sheet. Okay, that is showing the secondary structure. Means once the primary structure is formed, it form a coil like structure, that is the alpha helix, or it forms a sheet like structure, that is the beta pleated sheet like structure, right? So these two are the two different forms of the secondary structure. Now This secondary structure get folded in a different form. It undergoes the coiling and folding and form a three-dimensional shape. Three-dimensional shape, and that is referred as a tertiary structure of protein. Right? Now, these coils and folds are arranged to hide the non-polar amino acid. Now, they are folding in such that there should not be any non-polar amino acids, okay, and expose the polar side chain. So the polar side chain should be exposed and non-polar should be arranged in such a way that they should be in inner folding. Now, the tertiary structure is held together by the weak bond formed between the various parts of the polypeptide chain. So there are some weak bonds, van der Waals forces. Okay, so this forces are helping to form this folding of the or coiling of the polypeptide, right? So the helical polypeptide chain undergoes the coiling and folding, means secondary structure undergoes the coiling and folding to form a complex three-dimensional shape, and that is referred to as a tertiary structure of protein. Okay, so this is about the tertiary structure of protein. Now let us see the third question. Okay, find and write down the structure of 10 interesting small molecular weight biomolecules. So you have to tell about the 10 different, okay, interesting small molecular weight biomolecules like micromolecules, and find there is any industry which manufactures the compounds by isolation. Find out who are the buyers. Okay, so here we can see the first is the adenosine. So this is the adenosine. Okay, that is the nucleoside. You can see this is the sugar, ribose sugar, and this deoxyribose sugar, and here you can see the adenine. So this is the adenosine. Okay, the next one is the thymidine. Okay, uh, here again the ribose sugar is there, and you can see this thymine. Okay, nitrogenous base joined together to form the thymidine. Again, this is the nucleoside. And here is the third one is the sucrose. Sucrose is a sweet sugar. It is a sweet sugar. It is used for the sweetness. So here you can see two sugar monomers of the glucose unit unite together to form the sucrose. Okay. Okay, here you can see sucrose is this, one is the glucose and one is the fructose, whereas in the case of maltose you see the two glucose molecules unite together by the glycosidic bond. Okay, the bond in between the two glucose unit, that is the glycosidic bond you see, and it is forming the maltose disaccharide. These are the examples of the disaccharide, sucrose and maltose. Okay. Okay. Next is the lactose, a lactic acid, right? The conversion of this lactose into the lactic acid. And here again, you can see the two, this is a glucose unit, and this is forming the uh, disaccharide, that is the lactose that is present in the milk. Okay. Now, this is the ribose sugar, 5 you can see 5 carbon is there, 1, 2, 3, 4, and this is the fifth one. Okay, so this is the ribose sugar. Next is the DNA. You can see the structure of the DNA. So this is the ribose sugar. Okay, and with the ribose sugar you can see this adenine is joined here. Adenine is joined here, and this is the phosphate group. So this is one nucleotide. Okay, so nucleotide is formed by the nitrogenous base, sugar molecule, and the phosphate group. When joined together, it forms the nucleotide. Now, this is the another nucleotide, right? Thymine is there, ribose sugar, and deoxyribose sugar, and this is the phosphate group. So, and the third one is the cytosine. Cytosine is now joined with the another sugar molecule, and the phosphate group is there. Now, you can see this phosphate and ribose alternatively, right? And this nitrogenous base is inside. So this is forming the backbone of the DNA, and you can see this is the 5 prime to 3 prime. This is the orientation, or you can say the two polar ends of the DNA, right? 5 prime to 3 prime. And this phosphate and deoxyribose sugar, okay? These are forming this is the forming the backbone of the DNA, right? And the nitrogenous base is towards the inner side. So this is the structure of DNA, single helix. Now here comes the RNA, same thing. But what is the difference here? This is the ribose sugar. This is not deoxyribose. In the case of RNA, there is a ribose sugar, whereas in the case of DNA, the sugar is deoxyribose. So again, here you can see the 5 prime to 3 prime, that is showing the polarity, right? And you can see the phosphate group and the ribose sugar, phosphate, ribose, phosphate. So like this, it is the alternating phosphate and ribose sugar. And again, in the case of RNA, this is making the backbone. Now what you see here, you can see adenine, guanine, and uracil. Now, in the case of thymine, okay, you can see here the uracil, right, instead of thymine. So if it is A here, then its complementary will be A here, then its complementary will be U. Okay. Next is the glycerol. Okay, glycerol. So glycerol is CH2OH, CHOH, and CH2OH. 3 hydroxyl group is attached to the chain of propane, right? So here you can see the glycerol molecule. Now, the question is B, the compound, the manufacturer, and the buyer, right? So starch product, okay? Starch product manufacturer is one of the example is Kosha Impacts Private Limited, okay? Liquid glucose, that is the Marudhar Apparels, and various enzymes such as amylase, proteases, and cellulase, that is the MAP India Limited. So these are some of the manufacturer, and these are some of the uh, biomolecules which are synthesized in these manufacturers. And who are the buyers? Research laboratories, because the starch products, liquid glucose, various enzymes such as amylase, proteases, and cellulase, these are used in the different research projects, right? So there are different research laboratories that uses these compounds. Educational institute also required these compounds, and some of the industries which uses the biomolecules as the precursor of making the other products, right? So they require these biomolecules as well. So these are the table. This is the table that's showing the compounds, the manufacturer name, and also who are the buyers of these compounds. Moving to our next question, find out and make a list of proteins used as therapeutic agents. There are so many proteins that are used as therapeutic agents, right? So you have to make a list of protein that are used as a therapeutic agents. Find other applications of protein, example, cosmetics. Okay, so let us see the answer. Proteins that are used as a therapeutic agents are as follows. Thrombin and fibrinogen. You all know that for the blood clotting, there is a requirement of these uh, proteins, right? These are the proteins that are present or formed at the time of blood clotting in the blood, right? So thrombin and fibrinogen, they are helping in the blood clotting. So this is very, very important, and this is one of the use in the as a therapeutic agent, proteins that are used as a therapeutic agents. So thrombin and fibrinogen, they help in blood clotting. Antigen, right? So it help in blood transfusion, because antigen antibody reactivity is shown before the blood has to be transfused to a person, one person to the another. So there is a testing of both the donor and the buyer. So once the blood group is same, then only the transfusion of the blood is done. And for this, antigen antibody test is done, right? Next is the insulin. It help in maintaining the blood glucose level in the body. Now, this is the very, very important one of the mixed organ, that is the pancreas, right? That synthesizes hormone as well as the enzymes, right? So that is why it is called as a mixed gland, right? It is secreting both the type of secretion like hormones as well as the enzymes. So it helps to maintain the blood glucose level, because insulin is the hormone that allows the glucose to move inside the cell, and only then the oxidation of the glucose occur, and then it releases the energy. So what happens when the insulin is released in the blood? Okay, the glucose molecules get encapsulated, and then only it can get enter inside it. If the glucose is present outside the cell, okay, it will not move inside the cell unless it get encapsulated with the insulin. So insulin is, in that case, it's very, very important, and it is also used as a therapeutic agent, right? Next is the renin. It helps in osmoregulation. It helps in maintaining the uh, balance of the water in the body. So proteins are also commonly used as the manufacture of cosmetics, toxins, and biological buffers. So not only the renin is normally used, synthesized in the body as used in the osmoregulation, but it is also used as a manufacture to manufacture some of the cosmetics, because in the manufacturing some of the cosmetics, this rannin is used. It is also used as a toxins and other biological buffers. Okay, so moving to our next question, fifth question says, explain the composition of triglyceride. What is the composition of triglyceride? So let us see the answer. Okay, so the triglyceride is a glyceride which is formed from the single molecule of glycerol. We have just seen the structure of the glycerol: CH2OH, CHOH, and CH2OH, right? So 3OH group is attached here, right, with the single, that is the glycerol, esterified with 3 fatty acid. Now there is a esterification reaction. The acid react with the alcohol group, and it is forming the ester group, right? So that is why it is called as esterification reaction. So esterified with 3 fatty acid, and it is mainly present in the vegetable oils and the animal fat, right? So here you can see the 3R group, okay, and ester bond is here. One ester bond is this, this, and this, right? So this is showing the esterification. That is why it is called as esterified, right? So three acid group, fatty acid group, get attached with the glycerol molecules to forms the fat. Now, structure of the triglycerides. The general chemical formula of triglyceride. So R2COO, CH2CH, OCR1, CH2OOCR3. So here you can see R1, R2, and R3. Three different R, right? So where R1 and R2, 3 are, these are the fatty acid. Three different fatty acid is attached here, right? So three fatty acids can be same or that can be different. If it is same, then you can write it as R1, R1, and R1. And if it is all the three are different, then it can be written as R1, R2, and R3. Okay. Okay. So here comes our next question. Can you attempt building models of biomolecules using commercially available atomic model, ball and stick models? Okay. So ball and stick model are the 3D molecular models that can be used to describe the structure of biomolecules. Okay, so this is a way to, you can say, the description to describe the structure of any biomolecules. For this, ball and stick models, okay, that is showing the 3D molecular model of any biomolecules. That is used to describe how to describe the biomolecules. Okay, so in ball and stick model, the atoms are represented as balls. So here you can see in the figure, these atoms are represented as ball, right? So hydrogen, oxygen, carbon, and it is representing in the different colors, right? So in ball and socket stick model, the atoms are represented as the balls, whereas the bonds that hold the atoms are represented by the stick. So bonding is here represented as a sticks, whereas the atoms are represented as a ball. Okay, so double and triple bond are represented by the springs that are formed curved connections between the balls, right? So double and triple bond you can see with the help of uh, that is represented by the springs that are curved connections between the ball. Okay, the size and color of various atoms are different and are depicted by the relative size of the ball. So the size is also different. The color that is showing a particular atom is also different. So you can see the variation in the size and color of the various atom. You can see the bonding, right? Okay, the double bond, single bond, triple bond, that also you can be visualized with this ball and stick model. Okay, so it is the fundamental and a common model of representing the biomolecular structure. This is the way to represent any type of biomolecule, right? And this is the one of the most fundamental and a common model to represent any type of biomolecules. Okay, so in the above ball and stick model of the D-glucose, okay, uh, dextro glucose, that is showing the this stereotypic orientation. So the oxygen atom are represented by the red ball. So here in the figure you can see wherever there is an oxygen, it is represented as a red ball. Okay, and hydrogen atoms by the blue balls. The hydrogen is represented as the blue balls, whereas the carbon atoms are represented by the gray balls. Okay, so this is the way to represent, and this by this showing the different colors, you can easily visualize the atoms. Okay, by their color only, and the size of the ball also varies, showing representing the size of the atom. Okay. Now next question is here. Draw the structure of amino acid alanine. So you have to draw the structure of alanine. So here is the structure of alanine. So you can see here NH2 group is here. You can see OH group is here. O, and this is the carbon, right? Carbon is here, showing the double bond with O, single bond with OH this, and another with the NH2 group, right? So this is the structure of alanine. Okay, so next question. What are gums made of? Is Fevicol different? So the gums that is used as an adhesive, it is asking about that what are the gums made up of? So let us see. Gums are the heteropolysaccharides. Okay, these are chemically, if you see, then it is a heteropolysaccharide, right? The monomers are different. That is why it is called as hetero. If it is a same type of monomer, then it could be called as a homopolysaccharide. But here the monomers are different. So they are made up of two or more different types of monosaccharide. Now you can understand why it got its name heteropolysaccharide. Okay, because the monosaccharide used to form this polysaccharide are either it could be of two or more different types of monosaccharides. Now on the other hand, Fevicol is a polyvinyl alcohol, PVA glue. It is not a polysaccharide, right? So this Fevicol is a different chemical that is called as a polyvinyl alcohol, right? So this is a PVA glue and it is not a polysaccharide, whereas your gums are basically the polysaccharides. So this is the difference between the chemically, though both are used for like adhesive, right? Gums and Fevicol, but If you see the chemical based, then it is a heteropolysaccharides. The gums are heteropolysaccharide, whereas a fevicol is a polyvinyl alcohol, which is not a polysaccharide. So this is the difference between the gums and fevicol. Okay. Okay. So let us move to our next question. Find out a qualitative test for proteins, fats, oils, amino acids, and test any fruit juice like saliva, sweet, and urine for them. So this is one of the very, very important question of this chapter. Test of for protein. Biuret test. If the biuret reagent is added to a protein, then color of the reagent changes from light blue to the purple. Okay, the color, that is the biuret, that is you can say the reagent. Its original color is light blue. Okay, and if you are given any sample that you have to test whether this sample is having the protein in it or not, then you can take that sample and put some biurate reagent in that. Now when you are putting this biurate reagent, that is in the light blue in color, but when it react with the protein, it changes light blue to purple color. So this shows the positive protein test. Okay. Moving to the next type, that is the test for the fats and oils. So grease or solubility test is used. Okay. If it is substance is having the grease, oils and fat, then you put a butter paper over it and you can see the marks of the oils and fats over it. So that is the one of the test. Okay. Next, test for amino acid. For the test of amino acid, you can use ninhydrin solution or the ninhydrin reagent. Okay, so if a ninhydrin reagent is added to the solution, then the colorless solution changes to pink, blue, or purple depending upon the amino acid. Okay, so ninhydrin reagent, that is colorless solution. Okay, it is not having any color, it is a colorless solution, but when you add to a certain amino acids, it can change to pink, it can change to blue, it can change to purple depending upon what type of amino acid it contains, right? So this is for the test for the amino acids. Okay. Now, next is the different fruit juices. Okay. Fruit juices. And this is the second column showing the name of the test of biuret test, procedure, result, and inference. So, biuret test, when you do fruit juice and you add biuret test reagent, then color changes from light blue to purple. Again, it is showing the change in the color, that is the light blue to purple. The protein is present. Okay. Grease test. To a brown paper, add a few drops of fruit juice. No translucent spot. Then fats and oils are absent and are negligible amount. Okay. Next is the ninhydrin test. Take a fruit juice, add the ninhydrin reagent, boil for 5 minutes. Colorless solution changes to pink, blue, or the purple color. Then amino acids are present in that. Okay. Next, you take the saliva. You take the saliva in the test tube and you do this biuret test. Saliva added to the biuret reagent, then the color changes from light blue to purple. Proteins are present. So your enzymes are basically the ptyalin enzyme. That is salivary amylase. That is the chemical nature of it is the protein. So it will gives you the positive protein test. Okay. Grease test. On a brown paper, add a drop of saliva. No translucent spot is there. So fats and oils are totally absent. Ninhydrin test. Saliva, ninhydrin reagent, boil for 5 minutes. Colorless solution changes to pink, blue, or purple color because again, the protein is there, right? Enzymes. And when you heat it, it is broken down into their respective amino acid. So it will gives you the ninhydrin test positive. So colorless solution can change to the pink, blue, and purple depending upon the type of amino acid it is having. So amino acids are present in that case. Sweat. For sweat, you again do this biurate test. Sweat plus biurate reagent. No color change. In this case, you will not see any color change. So the proteins are absent. Here, the proteins are absent. Solubility test. Sweat plus water. You will take the sweat and water. Oily appearance. So fats and oils may be present. Ninhydrin test. You will take the sweat and ninhydrin reagent, boil it for the 5 minutes. No color change in the solution you can see, and it has remained the colorless. So amino acids are totally absent. Next is you will take some urine. Okay. So you go for this biurate test. Few drops of urine plus biurate reagent is taken in the test tube, and color changes from light blue to purple. So some proteins are present in the urine. Solubility test. Few drops of urine plus water, little bit of oily appearance. So fat may or may not be. Sometimes it is not present, but sometimes it may be present. Ninhydrin test. Few drops of urine plus ninhydrin reagent and boil for 5 minutes. Colorless solution changes to pink, blue, or purple color depending upon the type of amino acid it is having. So amino acids are present in that. So moving to our next question. Find out how much cellulose is made by all the plants in the biosphere and compare it with how much of the paper is manufactured by men, and hence what is the consumption of the plant material by men annually. What a loss of vegetation. So approximately 100 billions tons of cellulose are made per year by all the plants in the biosphere. So this is one of the data that is showing all the plant that is present in the biosphere. It is approximately 100 billions of tons of cellulose that are used every year by the plants. Okay. And 17 full grown trees to make 1 ton of paper. And for 1 ton of paper, you require 17 full grown trees, right? So trees are also useful to fulfill the requirements of men, such as timber, food, medicine, etc. It is not only giving you the paper, it is giving you the timber, wood, okay, the food materials, the medicines. We are getting so many things, and not only this, it is providing you oxygen to breathe out. Hence, it is difficult to calculate the annual consumption of plant material by them. Right? It is very, very difficult to make an assumptions, or to make a list that how much the plants are used for the making of the paper. Right? Describe the important properties of enzymes. So here comes the last question of this chapter. Describe the important properties of enzyme. So going to the answer, properties of enzyme. The first property is enzymes are complex macromolecule with high molecular weight. So this enzyme is a complex macromolecule with a high molecular weight. Second, they catalyze the biochemical reactions in a cell. So when they are present in the cell, they are helpful to catalyze the different biochemical reactions. So they help in the breakdown of large molecules into the smaller molecules, or bring together two smaller molecules to form a larger molecule. So sometimes there are different molecules that are broken down to make it a simpler forms, whereas two simpler molecules can join together to form another complex molecule. So both the type of reactions are taking place inside the cells. Okay, the catabolic and anabolic, both the reactions. And for every reaction, there is a requirement of a certain enzymes, right? So these enzymes are very, very important to take out all these types of reactions. Enzymes do not start a reaction, however, they are helping accelerating it. In the absence of these enzymes, the reactions are going on, but at a very slow pace. So it accelerates Okay, it is required to accelerate the rate of reaction. So enzymes are not used to start any reactions, but yes, it can accelerate the rate of reaction. Enzymes affect the rate of biochemical reaction and not the direction. Okay, the direction is not important and it is not like governed by the enzyme, but yes, the rate of the biomolecule reactions are controlled by the enzymes. Okay. Next, most of the enzymes have high turnover number. So turnover number of an enzyme is the number of molecules of a substance that is acted upon by an enzyme per minute. In 1 minute, how much you can say the substrate is changing to the product. So that gives you the turnover number. So high turnover number of enzymes increases the efficiency of reactions, right? So if the turnover number is more and more, then the efficiency of the reaction is quite high. Enzymes are specific in action. So they are very specific, then they can take care of this particular type of reaction. Okay. Enzymatic activity decreases with the increase in temperature. When the temperature is increased, the activity of this enzymatic reactions goes down. Okay. They show maximum activity at an optimum pH 6 to 8. So this is one of the pH at which the generally the enzymes are showing its you can say the maximum activity 6 to 8. The velocity of enzyme increases with the increase in the substrate concentration and then ultimately reaches to the maximum velocity. So if you keep on adding the enzymes, okay, you keep on adding the substrate, okay, the rate and the concentration of the substrate is added, okay, then it increases the rate of reaction and it reaches to the maximum velocity. Okay, so with this, we have completed this chapter. So thank you very much for attending this session. We will meet with a new chapter in another session. Till then, thank you very much.